right, today we are going to talk about cell structures. This is going to be, we're not going to spend a lot of time on any one structure because we'll talk about several structures as we move through the class. This is going to be a nice survey of the different structures that can be found within cells. Now, when we talk about the cell structure, cell organelles, and we talk about the cell, the animal cell, or the plant cell, or the prokaryote bacteria cell, we're talking generalized structures. So what I mean by that is that in most cases, animal cells will have this. In most cases, plant cells will have that. In most cases, bacteria cells are set up like this. There are differences between the cells. There's going to be cells that have a lot more mitochondria than other cells. There are going to be cells that have a lot more lysosomes than other cells because cells are extremely specialized. What a liver cell needs and uses is very different from what a muscle cell needs and uses. So we're talking about generalized structures, but it's important that you understand that these structures can be found in different types of cells and in different amounts in any of those cells. Okay, oops. Here we go. So we're going to start with the cell theory. Now the cell theory, as you read in your book, is something that was discovered over time. There were several scientists that played big roles in developing the cell theory, but with the cell theory, we came away with 3 precepts, 3 important things. All right, the understanding that all living things are made of cells. When we talk about characteristics of life in chapter 1, we talked about if it doesn't have a cell, it's not living. All right. All cells come from pre-existing cells. There is no spontaneous generation. There has to be an original cell there in order to have more cells. We may not know how the original cell got there, but there has to be a cell in order to have more cells. So once again, characteristics of life, there has to be reproduction there. Cells are the basic unit of structure and function. So understanding how a structure works at the cellular level helps us understand how that structure might work at an organism level. Understanding a function such as digestion at the cellular level helps us understand how it works at the organ level. So you can have many different types of specialized cells, and within the cells you can have many types of specialized structures. So what you see here is a nerve cell in the nasal area, and on this nerve cell you have cilia. And the cilia is an example of a specialized structure. They can be found on the surface of some cells, not all cells, some cells, such as the cells in your respiratory system, and they will move materials over those cells. So, for instance, they might be moving mucus up out of your bronchioles so that you can cough it when you feel sick. So that's just an example of a specialized cell structure. And we'll talk a little bit more about cilia later. You can see in this picture how cells can have very, very different shapes. We talk about human red blood cells kind of have a donut shape, right? So they're circular, and they have a biconcave where it's thinner in the middle than it is on the outside, and that allows for increased cellular diffusion so that we can move oxygen into that blood cell and take it where it needs to go within the body. Yeast cells. Yeast reproduce through a process called budding. So what you're seeing here is actually a new yeast cell being formed. A new yeast organism forms by growing out of the original yeast organism. And over here you have spinach cells. And spinach cells, we're talking about a plant. So they're going to have structures that red blood cells don't have, and they're going to have structures that yeast doesn't have, which is a fungus. So when we talk about types of cells, we have to talk about the difference between a prokaryote and a eukaryote. Now when we talk about the terms prokaryotic and eukaryotic, the kary right here has to do with the nucleus. So pro is before or first, right? So a prokaryotic cell is before, spell it right, nucleus. All right, before the nucleus. So prokaryotic cells do not have a nucleus. They haven't yet. They haven't. They don't have it. They just don't have it. When we talk about over time, prokaryotes are older organisms than eukaryotes, and so they were before the development of the nucleus in an organism. Eukaryotes, where U right here means true. So they have a true nucleus. So the first major difference between prokaryotic cells and eukaryotic cells is prokaryotic cells do not have a nucleus for their DNA. They have what's called a nucleoid region. A eukaryotic cell, their DNA is in the nucleus, right? The easy way to remember this is Y-O-U, are eukaryotic. You are eukaryotic. Your cells have nuclei. You are eukaryote. Most of your prokaryotes are single-celled or unicellular, like bacteria, while eukaryotes are larger than prokaryotes, and they are often multicellular. There are some single-celled eukaryotes, but the majority of the eukaryotic family, the majority of the eukaryotic group, is multicellular organisms. Prokaryotes, even though they're simple and they are very small, there is a lot of diversity. So there's a lot of different types of prokaryotes out there. Examples of these are bacteria or archaea. All right, so there's a wide variety of prokaryotes. They do important things for us. Sometimes they make us sick, but they also do extremely important things for us. Digestion would not happen without them. But classified as a prokaryote, does not have a nucleus, does not have membrane-bound organelles. You're basically looking at a cell membrane, cytoplasm, ribosomes, and DNA. Eukaryotes, they have organelles. They are organized, they are compartmentalized. Those organelles have specific functions. And because you have greater organization, because you have greater multiple different structures with multiple functions, eukaryotic cells tend to be more quote-unquote advanced. So they can accomplish and specialize a lot of different things. All of life on Earth, except for prokaryotes, are eukaryotes. Now, don't make the mistake of thinking because all of life other than prokaryotes are eukaryotes, that eukaryotic cells are the majority. There is so many different types of bacteria and archaea out there. So there are several different species of those organisms, just like there are several different species of eukaryotes. All right. Prokaryotes will have a peptidoglycan cell wall. So they have a cell wall. Peptidoglycan is what it's made of. When we talk about cell walls in plants, we talk about cellulose, but we're looking at peptidoglycan, which means it's made of a protein and a carbohydrate. So that peptid is your protein and glycan is going to be your carbohydrate. Most of them will have a capsule around them. All of them will have DNA found in what's called the nucleoid region. The nucleoid region is just simply the area within the prokaryote that you can generally find the DNA. It's not enclosed in a nucleus, it's just in a region. The DNA in a prokaryote is single, typically, there's your DNA, circular, and you tend to have a single strand of it. All right. Prokaryotes can have a flagellum. Flagellum allow for movement. They can have cilia, like we saw on the first page. They can have pili, and these pili will extend from prokaryote to prokaryote like a tube. So you'd have a prokaryote, and the pili would extend, and DNA can move along that tube. And then fimbriae. And fimbriae are used to help attach to host cells so that they can attach and have that environment to live in. There's a huge variety in size. When we're looking at prokaryotes like this bacteria, all right, so this is like on the tip of a pin right here. It's the least amount of magnification, and you get to the greatest amount of magnification. You get to start to see those individual organisms. So those are all bacteria. bacteria are going to range from here to here. So they're very small. Then when you move up, you start to see your plant and animal cells are right here. And then you have your eggs, and then us, and then the blue whale. All right, so you can see there is a very large difference between the smallest bacteria and the largest organisms. Prokaryotes tend to be 0.1 to 0.5 micrometers in diameter, so very small, while eukaryotic cells tend to be anywhere from 10 to 100 micrometers in diameter. There are some single-celled eukaryotes that are visible to the human eye, but not very many. Most of them are microscopic. The small size of the cell allows for the diffusion of ions and the diffusion of molecules into and out of the cell. And we're going to talk more specifically about moving materials into and out of the cell later. But when you are smaller in size, your volume, or what your cell needs in order to function in relationship to your surface area, is small. Which means if you're a cell, the only way you can get food and materials into your organism, and the only way you can get rid of waste out of your organism, is through that cell membrane. So it's important that my volume, my metabolism, is able to move materials in and out as needed. If I get too large, my volume increases a lot faster than my surface area does. So what happens in that case is now my metabolism has a much higher requirement, and my surface area will not move the materials in fast enough. So as cell sizes increase, the surface area to volume ratio decreases. If you get too large, you can't get enough materials in order to function. So that's one of the reasons why you don't see a lot of macroscopic cells, is because it's very hard for them to get what they need in order to survive, in order to take care of themselves. So this is a nice little diagram that just kind of reviews the differences between prokaryotes and eukaryotes. Eukaryotic cells, and we're going to spend most of our time today on eukaryotic cells for several reasons, because you are eukaryotic, so it's always nice to learn about ourselves and the organisms around us, like our puppers and our cats and our dog and our horses and stuff like that. So it's nice to learn about the organisms that surround us, but also because eukaryotes are more complex. They have structures called organelles. And so when we learn about eukaryotic cells, there's just more to learn about. So they have membrane-bound nucleus that contains the DNA. If you remember, prokaryotes had a nucleoid region. They have membrane-bound organelles like endoplasmic reticulum and mitochondria and chloroplast and Golgi apparatus. And these membrane-bound organelles perform specific functions for those cells. Eukaryotes also have several chromosomes. Humans have 46. All right, so when you think about that bacteria, that E. coli bacteria, it just has the one singular circle chromosome. Humans, we have 46. So there's a lot of differences between us and bacteria. When we talk about eukaryotic cells, there's a lot of structures that are found in all eukaryotic cells. We're going to focus on these 5 structures, and then we're going to quickly hit some other structures. And we're also going to focus on the organelles that are important for protein synthesis, for making proteins, because proteins are such an essential portion of living organisms. So the 5 major structures: the nucleus. This is the defining characteristic of a eukaryote. If you don't have a nucleus, you're not a true eukaryote. Membrane-lined compartment. It helps to enclose the DNA. DNA doesn't leave the nucleus. It stays in there protected. The nucleus produces RNA, and that RNA will go out into the cytoplasm for protein synthesis. Organelles are highly organized structures within the cell. They all have specific functions. Cytosol, when we think about the cell and the material that's inside of the cell, we think about cytoplasm. Cytosol is the protein, that gel-like fluid that's inside the cell that holds the organelles. If we're just talking about the fluid-like substance, we're talking about cytosol. If we talk about the fluid-like substance plus the organelles, you're talking about the cytoplasm. And then the cytoskeleton. The cytoskeleton is pretty interesting because it's made of several different types of proteins, and they have several different functions. And even though cells do not have like a skeleton like we have, or animal cells don't have a cell wall to give it structure, that cytoskeleton plays a major role in protecting the structure of the cell and also allowing for movement of materials around the cell. And then last but not least is the plasma membrane. Now, we're not going to hit the plasma membrane super hard today because when we talk about the cell membrane later, we'll get into all the weeds surrounding that. But the plasma membrane lines the cell. It gives it that boundary and it controls what can enter and leave. The plasma membrane is made up of phospholipids. Now, if you remember from our macromolecules lecture, a phospholipid has a phosphoglycerol head and then 2 fatty acids on it. And then the top of the phospholipid is hydrophilic. Ooh, that is not easy to read. Hold on. That's better. Hydrophilic, which means it will be around water. And the bottom part is hydrophobic. Now, the phospholipid bilayer is what makes up that plasma membrane. And if you look at this, you can see that the heads right here are on the outside of the cell. So these heads are going to be exposed to the extracellular fluid, which is going to have water in it. And they're also, if you see, on the inside of the cell. And they're going to be exposed to intercellular fluid, which will have water in it. So the phospholipids are lined up in a way that the heads are exposed to water substances with water in it, but these little tails right here are protected. And because of that, and we'll talk more later, it determines what can enter and leave the cell. So it's very controlling as to what can enter and leave. It's picky. It separates the internal contents, the organelles of the cell, from that environment. And we're glad that it's picky because we don't want it to let in things that it shouldn't. We talked a little bit about this, cytoplasm versus cytosol. The cytoplasm is the area outside the nucleus and inside the plasma membrane that contains organelles. It also contains ions, glucose, proteins, amino acids, things that the cell needs in order to function correctly. This is where your metabolism of the cell is occurring. So we're breaking down materials and we're building materials that the cell needs. And then the cytosol is what is left after you move all of the structures. So we take out the Golgi apparatus and the lysosomes and all those organelles. What is left, that gel-like fluid, is your cytosol. All right, so specifically talking about animal cells. Animal cells, or the typical animal cell, is spherical. Now, not all animal cells are spherical. You can have animal cells that are column-like in shape. You can have animal cells that are flat in shape. You can have animal cells that are kind of cube in shape. And they can be spherical in shape. You talk about like your red blood cells. They're that spherical shape. They are surrounded by and linked to like cells. So cells in animals, such as yourself, tend to make tissues. So they join together with other similar cells. They're found in a watery fluid known as extracellular fluid, and that is the fluid through which diffusion is occurring. They are approximately 25 micrometers in diameter. So they are much larger than a prokaryotic cell. So when we talk about organelles in this animal cell, specifically in the animal cell. Now this occurs also in plants, but just because we're going through animal cells, we're going to follow protein production and talk about all of the organelles that are essential in forming those proteins that we need in order to survive. So this starts at the nucleus. The reason we're starting at the nucleus is because the nucleus has the DNA, which are the instructions needed for protein production. It is the code that lets the cells know how to build those polypeptides, how to string the amino acids together and make a protein. The nucleus also directs the synthesis of ribosomes. and the proteins that are needed for ribosomes. It is surrounded by a nuclear envelope, and it is just a membrane that surrounds the nucleus, very similar to how the plasma membrane surrounds the cell. It controls what enters and leaves that nucleus. And the nucleus is also extremely important in cell replication because that DNA inside the nucleus has to duplicate and then split into two new nuclei. So the nucleus is extremely important. It has the code, it controls what is being produced, and it allows for replication. Now, when we talk about the code, we're talking about DNA, the code. Remember, DNA is a type of nucleic acid. It comes in different forms. Now, it's all DNA, but the difference is the shape and whether or not the DNA is stretched out so that it can be used or compacted so that it can be sorted. So chromosomes are structures in the nucleus made up of DNA. Like I said earlier, prokaryotes have one, eukaryotes have many. Humans have, do you remember? Humans have 46. You only see chromosomes when a cell is getting ready to divide. So this chromosomes are compacted. All right, so it's like if you have a bunch of electrical cords and you need to move them, they're a lot easier to move if you get them all wound up into a bundle as opposed to just trying to move all of those electrical cords from one place to another without trying to contain them at all. That's what is happening with chromosomes. The chromosomes are compacted, they're twisted really tight so that they can be sorted and shifted and moved around. Now, so that is during division. Chromatin, it's still the same DNA as the chromosome, but it's in the unwound form. And so that is basically, it's open, it's stretched out so that we can access the code that's within it, so that that code can be used to make proteins. So most of the time, the DNA in your cells are in chromatin form or stretched out form, that pile of electrical cords that are unwound and being used. The only time you see the chromosome or the chromosome is your chromosome is your X form. When we talk about DNA and it's kind of that X shape, that's the chromosome form. It's only found during cell division. Most of the time, it's just kind of stretched out so that we can access it and we can use it. Now the nucleolus is inside the nucleus. So if this is your nucleus, there's going to be a darker cell or darker portion in the middle of it. Like this is your cell right here. This is your cell. And then your nucleus and then your nucleolus. The condensed portion in the middle. This is where ribosomes are made and also RNA. Ribosomes are made of ribosomal or rRNA, and these are made in the nucleolus. Ribosomes are extremely important because they allow us to make proteins. So you have your nucleus, you have your nucleolus, and then you have your nuclear membrane. Now notice in the nuclear membrane are pores. And what happens during protein synthesis, DNA is used to make RNA, and that RNA goes through the pores into the cell. The type of RNA that is taking the message of the DNA from the nucleus to the ribosomes is known as messenger RNA. So this is the portion of information that is actually being used to build a protein. So basically what is happening is the DNA is the code, and then the code has to be translated into something that we can use, and that is messenger RNA. And the messenger RNA leaves the nucleus and it goes to the ribosomes, moves to the ribosomes so that protein synthesis can occur. Now at ribosomes, you have protein production. So we are finally making proteins. So that is the function of ribosomes. It's the site of protein production. Made up of two subunits, large and small. So you have a small subunit, and then a large subunit is kind of attached to it. It's the only organelle found in both prokaryotes and eukaryotes. Proteins are essential for function. Prokaryotes need proteins just as much as we need proteins in eukaryotic cells. They can be found in the cytoplasm as free ribosomes or attached to the plasma membrane or attached to the endoplasmic reticulum. And those are called bound ribosomes. When we have ribosomes attached to the endoplasmic reticulum, you have rough endoplasmic reticulum. You might see this abbreviated RER. All right, so just remember it is rough because of ribosomes. It basically, when you look at it under a microscope, it looks bumpy, so it's rough. This is a folded continuation of the nuclear envelope. So you have your nucleus, and then an extension of that is going to be your rough ER, and it's going to have ribosomes bound to it. That is the most beautiful drawing I've ever made. And the job of those ribosomes is to fold protein shapes. So basically they're finishing off proteins so that they can be used. The proteins in rough endoplasmic reticulum are typically going to be used in the cell membrane. So they will be shipped to the cell membrane where they can be used there, or shipped out of the cell so that they can be used in other places. These are found, you find a lot of rough endoplasmic reticulum in cells that secrete a lot of proteins, such as liver. So if your job is to make a lot of protein, then you're going to have a lot of rough ER. Secreted protein, because it's being transported out. So rough ER is helping to make proteins that are going to be transported out of the cells. And there's an example. You have your mRNA that's made from the nucleus. You have your ribosome is where protein production occurs, and you can see it looks like it's pooping out a little green protein, and then that protein is going to be put in a vesicle, and that vesicle will ship it out. Not all ribosomes are found on the rough endoplasmic reticulum. You can have free ribosomes, and these are just found in the cytosol, so they're found within the cell, and free ribosomes make proteins that will be used in the cytoplasm or in the nucleus. So these proteins stay in the cell, while rough ER shipped out. All right, so how are these proteins shipped out? How are materials shipped around the cell and out of the cell? And they are shipped through transport vesicles, and they are literally sacs. They are bags made of membrane that will bud off of organelles like rough endoplasmic reticulum or smooth endoplasmic reticulum or Golgi apparatus. They are literally membrane bags, and they carry things around the cell. They might carry them to the cell membrane so that the material can be pushed out. They might carry them to another organelle, but vesicles are membrane bags, and they carry things around. Now, the Golgi apparatus is an interesting little creature. It is another network of membranes. It kind of looks like stacked pancakes. All right, and it distributes those proteins from the rough endoplasmic reticulum. It has an in that accepts vesicles, and it has an out that sends out vesicles. So it receives material from the endoplasmic reticulum, rough endoplasmic reticulum. We get to the Golgi. It messes with it. It sorts it into different vesicles. It tags it with carbohydrates. It packages it, and then it distributes it to different locations. So it might go to other parts of the cell. It might go to the plasma membrane. But it's receiving and it's shipping. A common way of explaining it is it's like the UPS of your cell. It brings those vesicles in, it ships, packages, it ships, and then it sends them back out. It has a receiving side, so this is the receiving side is cis. And then it has a transport side, which is trans. That's easy to remember because you have transporting is the trans side. That's the exit side. All right, so we're receiving materials in to the cyst side. That material gets manipulated while it's in the Golgi. It gets put back into a vesicle right here, and then it gets shipped, in this case, out of the cell. And this is a micrograph. It's pretty cool. You can see all of those layers right through there. Cis, so we're going this way, and trans, we're going that way. And you can see coming off of this Golgi apparatus, all these little bubbles, those are vesicles that are coming off those Golgi apparatus. So once we get it all packaged into those vesicles at the Golgi, it heads to the cell membrane, and it will fuse to the membrane and release into extracellular fluid. So you have a membrane, and your vesicle is moving that way. It will actually fuse to the membrane or attach, and then what it'll do is it will open that vesicle up. It'll open it up. So if you got blue dots in here, and those blue dots, and then eventually you no longer have a vesicle, and those materials are outside the cell. All right, so what else? What else does the cell have other than organelles directly involved in protein synthesis? There are still other organelles. They still have important functions. We can't ignore them. So let's talk about those real quick. The smooth endoplasmic reticulum is another series of folded membranes, much like the rough endoplasmic reticulum, but the smooth does not have ribosomes. It is ribosome free. That's why they call it smooth, because it doesn't have those bumps on it. They function in lipid production and detox. So this is another type of organelle that you're going to see a lot of in the liver. They are away from the nucleus, so they're not connected to the nucleus like the rough endoplasmic reticulum. They don't have ribosomes. They help to assemble fats. These fats can be used in the cell membrane. They help to assemble testosterone and estrogen and things like that. And they also help to detox, like alcohols and stuff from your body. Lysosomes are similar to vesicles in that they are just like a bag, but a lysosome contains enzymes, and those enzymes are breaking things down. So breaking materials down. So they return those smaller molecules to the cytosol for reuse. So we're doing some recycling here. That is hard to read, I apologize. Let's go with a color that can read. We recycle so that we can reuse those materials. And sometimes membrane-bound vesicles fuse to lysosomes, and those lysosomes will insert enzymes into those membrane-bound vesicles to break things down so that you're getting rid of waste. Think of the lysosomes as like a digester. They digest old cell parts, old cell materials, or garbage disposal. Because they break things down. You can see you have your lysosome right here that has those enzymes, and you have an old mitochondria right here. He's done a good job giving us energy, but his lifespan is over. So they will join together, and the enzymes from the lysosome go in and break down that mitochondria. What we can use from the mitochondria goes back to the cytoplasm, and then that can be used to build other materials, and what cannot be used gets pushed out as waste. Peroxisomes are similar to lysosomes. They carry out oxidation reactions that break down specifically fatty acids and amino acids. So peroxisomes are a specific organelle. They break down only specific things. And one of those things is poisons. So we're breaking down fatty acids and amino acids, very specific in what we break down. Mitochondria, we'll talk more about mitochondria when we talk about cellular respiration, because it's essential for cellular respiration, but it's that primary site of energy conversion in eukaryotic cells. This is the organelle that takes us from glucose to ATP. So it's extremely important. The amount of mitochondria within a cell can vary according to energy needs. If we need a lot of energy, we're going to have more mitochondria. So for instance, our muscles, our skeletal muscles, require a lot of energy for us to work. And they have a high amount of mitochondria within them. They are oval shaped. They have a double membrane, so 2 membranes, and they have their own ribosomes and their own DNA. In fact, there's a hypothesis out there. So this is kind of our oval shape. It has a double membrane on the outside. It has a membrane on the inside. It's kind of a funky looking mitochondria. And it will have DNA and ribosomes. And there's a theory out there that at one time in our past, mitochondria were their own organism, and eukaryotes co-opted them. It was a symbiotic relationship that in symbiosis means that both organisms are benefiting or are interacting in a mutualistic relationship. They both benefit. So there was a benefit somewhere in the past where those mitochondria gained maybe protection or environment or a constant source of glucose when they were co-opted into the eukaryotic cell, and the eukaryotic cell gained ATP, energy production. So there's a theory out there. It's called the endosymbiotic theory about mitochondria, and also actually about chloroplasts. Because mitochondria is the only organelle that has its own DNA in animals. If you're talking about plants, mitochondria and chloroplasts have its own DNA. You might have heard of mitochondrial DNA. It's inherited through the mother's side. All right, and that is a diagram of a mitochondria. Cytoskeleton is a network of protein fibers. It's used for structure, it helps cells move, and it helps to move materials within the cell. There are 3 types, and they're named according to size, with microfilaments being the smallest and microtubules being the largest, and the intermediate is in between. And what you're seeing when you look in this picture, this phosphorescent picture, it's really cool. You can see all this green right here. All the green that you're seeing is cytoskeleton within cells. All right, so talking first things first, microfilaments are your smallest one. They're made of actin. Actin is a protein. If you've studied anatomy at all in the past, actin is an extremely important protein used in muscle contraction. It's used for structure and support in the cell and allows cells to have movement so that the cell can extend. Like you see this cell right here has an extension, and it looks like it's hunting this little bacteria right there. And that cell, that actin, will move and extend out, and it allows the cell to move towards, in this case, its prey. Intermediate filaments are in between microfilaments and microtubules. They're the most permanent part of the cytoskeleton, so they're not going to change a whole lot, like the microfilaments moving towards something and the microtubules, which are essential in transport. These are mainly your structural filaments. They're going to stabilize the position of the nucleus and organelles and give it stability. It helps to resist, it's important for helping to resist changes in shape. And then microtubules. This is the largest cytoskeleton filament. It's tube-shaped, so tubules are tube-shaped. They're structural. They help determine shape. They help to resist compression or squeezing. And they serve as monorails for motor proteins. So if you're watching this, I highly suggest you download the PowerPoint and watch this GIF, because this little guy right here is a motor protein, this little dude right here, and he's attached to a vesicle, and he is literally walking down this microtubule, moving materials around the cell. It is amazing to watch because we don't think about this kind of function happening at such a microscopic level. So your major cytoskeleton components, your microfilaments, the thin actin, microtubules are thick, they're very thick, and then your intermediate filaments are in between microtubules and microfilaments. Now, we talk about cytoskeleton and we talk about microtubules and things like that. And that's very important because they allow for cell extensions. So we talked at the very beginning about cilia, and cilia are extensions like this. They're hair-like and they move back and forth. Oh, I messed up. They move back and forth, all right, in a wave-like pattern, and that helps to propel the cell through fluid if you're talking about like a bacteria. But if you're talking about a stabilized cell, like the cells in our respiratory system, it moves materials over the cell. So the example we used earlier is that these cilia are moving back and forth, and they're carrying, for instance, mucus up the respiratory tract so that it can be coughed out. Flagella is a long tail-like extension. We do have flagella within sperm. So that little flagella will move back and forth like a whip. It can also corkscrew. Some flagella can corkscrew, have a corkscrew motion. But it helps to propel the cells through fluid. So here's your cilia as a back and forth, and this is your corkscrew motion of your sperm. All right, and this is your cilia. And you can see there's some pollen looks like trapped in that cilia, and it will help to push it out. And then your sperm, basically a DNA delivery system here. The head of the sperm has chromosomes, not much else in the sperm, mainly just that DNA. And so it will deliver that DNA to the egg and allow for fertilization. Some really good videos. Once again, I strongly suggest that you download this PowerPoint and you watch your cilia movement and your flagella movement. All right, centrosome. Centrosome is something that you only see in animal cells. They think that it is essential. It is involved in cell division, but it is not essential for cell division. When they remove centrosomes out of the cells, cell division still occurs. So there's a little kind of fuzzy area as to what exactly a centrosome does. And you would think after studying cells for as long as we have that we would, have this down pat, but we don't, because they're extremely intricate and we're always learning more about cells. But they think that centrosomes have a role in cell division and they help to separate the chromosomes. So in cell division, you've got your chromosomes and you want to separate them out so that each new nucleus has a chromosome in it. And then they think that that's being done through spindle fibers, and the spindle fibers come from your centrosomes, which would be right here. All right. So let's talk specifically about organelles that are only found in plant cells. Now, some of the organelles that we've talked about in the animal cell section can also be found in plant cells, but let's specifically talk about the ones that are only found in plant cells. Starting with, here's your plant cell. We got chloroplast, a large central vacuole, and then a cell wall. That's what we're looking at. The central vacuole, which is this thing right here, is extremely large. And it is very important because it helps to maintain cell pressure. It stores nutrients. It helps with retention and breaking down waste, but it's extremely important for maintaining cell pressure. What we have to remember about plants is plants do not have a skeleton that allow them to stand up. They have cell walls and they have a central vacuole. So think of it kind of like the central vacuole being a balloon inside that cell. When the balloon is full, it's putting pressure on the cell walls and it is giving it more structure. When the balloon is empty, it is not putting pressure on the cell walls and it's not giving it as much structure. So the central vacuole is essential for helping maintain pressure within a cell, called specifically turgor pressure. 90% of a mature plant cell is filled with the central vacuole and the water that it contains. All right. The amount of water present helps to encourage movement of within and out of the cell, of the amount of solutes present. So the amount of ions and things like that help to encourage water to move into the cell continuously and give it that pressure. Now the pressure that we're talking about is called turgor pressure. And it just literally, when it is full, when the vacuole is full, it is pushing on the cell wall and it's giving the plant greater structure, and you see it standing up. When it is empty, or I don't even like the idea of empty, but it could just be low. It could still have water in it. It just doesn't have enough water in it to push it against the cell wall, and then you have a wilted plant. If we add water back into the system, so if we put water into this plant right here, the plant, using the roots, will uptake the water and distribute it to the cells, and then before you know it, it's standing straight up. The plant, like you can see this, and we've all seen wilted plants before, but if you've ever noticed or owned a peace lily, they are really good about telling you when they are thirsty. When they are thirsty, they are droopy as all get out. You put some water on that peace lily, within a couple hours, it looks fine and dandy again. And it all has to do with that water pushing, creating a pressure on the cell wall. The cell wall is an outer protective covering in plant cells. All plant cells have a cell membrane. So we still have a cell membrane in our plant cell, but the cell wall is a really thick outer membrane around that cell. And it is made of cellulose. In plant cells, it's made of cellulose. It's nearly always present in plant cells. You can find them in bacteria, fungi, and protists as well. The cell wall is the rule, not the exception. Animal cells are the ones that don't have cell walls. Most everything else does, and it gives us strength, and it gives us protection. It limits the amount of water absorption, so we don't get too much or we don't lose too much out of it. It is rigid and inflexible. It has pores in it, and materials can move through the cell wall. Several different types of cell walls in plants. So depending on the type of plant you can have different cell wall structure. There's a lot of metabolism happening in cell walls because it's controlling what's moving in and out. In plants, they are made of cellulose. If you remember, cellulose is a polysaccharide, so a complex carbohydrate. If you've heard the term fiber, whether you are talking about soluble fiber or insoluble fiber, we are talking about the cellulose that is found in cell walls. Cellulose can also be joined by a compound called ligand. And when we have ligand in there, that ligand acts like a rebar and increases the cell wall strength, and it gives you wood. So when you have cell walls that have ligand, we call that wood, like a tree or bark. All right, and this is a magnified view of bark. Chloroplasts are very important for plants because they are the site of photosynthesis. So this is where this is the organelle in which sunlight gets converted into glucose. All right, so this is how plants eat. It's the only way they can get energy. Then there's another process that occurs in the mitochondria called cellular respiration, where the glucose gets converted into ATP. Okay, so this is cellular respiration. And this up here is photosynthesis. All right, they are the food factories, chloroplasts, and therefore plants for most of the living world. So this is how the energy from the sun gets put into chemical form that we can then use and put it in a mechanical form for movement, for production of materials, so that life can exist. They harness that energy and they use water and carbon dioxide and they make glucose and give off oxygen as a waste. Now, it's weird to think that something that is such an important part of our life is a waste product for photosynthesis. But then plants will turn around and take that oxygen and the oxygen within the atmosphere and use it to power cellular respiration. Chloroplasts are very abundant in leaves. They have a pigment called chlorophyll, and that is green. And this is why we see lots of green leaves. They have an inner membrane and an outer membrane. Inside the chloroplast, I kind of need green, don't I? Let's see if I can find green. So if this is our chloroplast, inside the chloroplast, they have these button-like structures, sacs called thylakoids, and they come in stacks. And those stacked sacs are called granum. So a single one is a thylakoid. The whole shebang, the whole stack, is a granum. And this is surrounded by a fluid. All of this is surrounded by a fluid called stroma. Now inside chloroplast and inside the sacs, you have that chlorophyll, which is the pigment that collects that solar energy. So it's capturing that light energy, and that allows for the chloroplast to convert that into glucose. So here, we'll go back to red, we have a single thylakoid. Here we have a stack of thylakoids called a granum. All right, and you can see that inside the chloroplasts. All right, let's talk for a moment about cell communication. And this, I believe, finishes up our lecture. But cells communicate with each other. Now, they don't talk, they don't send texts or pass notes or use sign language, but they do send signals to each other, and they do communicate to each other. And they do that with different types of junctions. It's extremely important. So, for instance, heart contractions. Our heart cells are not innervated, which means not every single heart cell in your heart has a nerve attached to it telling it when to beat. What we have in our heart are nodes, and the nerve goes to the node, and the node sends out the information, and that information moves from cell to cell and then contracts. So the cells are passing that signal, not a single neuron. So they're passing it back and forth. So cell communication is extremely important. Most of our cells are organized into tissues, and those tissues allow the cells to have function together. And because they are functioning together, they also kind of need to be on the same page. They need to communicate. If we're going to work as a team, we need to communicate. So there's different ways that cells communicate with each other. A lot of it has to do with the extracellular matrix or materials outside the cell, and receptors that allow the cells to signal each other, kind of like how enzymes interact with their substrates. The receptors will signal other cells. So we have different types of junctions, and junctions are connections between cells. First, you have a tight junction. These are watertight seals between animal cells. They're held together by proteins, and that helps to keep leakage from occurring in between cells. So you have a junction that holds them so tight, nothing can move between them. And then you have a gap junction. And gap junctions have channels specifically so things can move in between them, such as electrical signals, such as signals for heart contraction. Desmosomes are found in animal cells, and these are spot welds that allow the cells to stay in a sheet, such as your skin cells. Skin cells. Skin cells will attach to each other and they form sheets or layers. And then we have plasmodesmata. Plasmodesmata is the only one that we're talking about has to do with plant cells. And they are channels between plant cells that allow for materials such as water and cytoplasm and ions to move from cell to cell. All right? So tight junctions, tight. Don't allow anything through animal cells. Gap junctions allow for movement of information such as nerve impulses. Desmosomes are spot welds that hold the cells together like sheets, and then plasmodesmata have channels that allow for movement of materials between plant cells. All right, so plasmodesmata right here allows for the movement of materials. Tight junctions really hold those cells together tight. And then you have desmosomes that are like spot welds that are holding them together. And then we have gap junctions, and a gap junction is a channel that allows for the movement of materials into and out of the cell. So once again, we've covered a lot of material very quickly in this lecture, but it's important that we understand how all of these organelles work, how they work together, and how they allow for a cell to function correctly. If you have any questions, give me a holler, shoot me an email, send me a remind text, or contact me on Blackboard. Have a good day.